Okay, so today we're going to be talking about operons. Operons are important because they control gene expression. So they allow organisms to conserve energy and resources by preventing the production of unnecessary proteins. They also ensure that proteins are only made when needed. So they do this through something called inducible and repressible operons, which I will get into in a second. So when we're looking at an operon's structure, it can be fairly simple. You have a regulatory gene, you have your DNA, you have your promoter, you have your operator, you have your genes that are going to get expressed later, and then you have your terminator. So you have your regulatory gene, which is going to code for a repressor, which I'll talk about in a second. You have your DNA. Your promoter is going to be where RNA polymerase is going to bind to. Similar to DNA replication, RNA polymerase is going to bind here, and it's going to get the go-ahead. If the operator doesn't have a repressor, it's going to get the go-ahead to transcribe these genes here. If there's a repressor here, RNA polymerase isn't going to be able to transcribe these genes, right? So kind of think of the operator as the green light and the red light at a traffic stop. So in terms of repressors, repressors have a DNA binding domain. This is a repressor right here. That's going to bind to the operator. If the repressor binds to the operator, like I said, then RNA polymerase is not going to be able to start transcription. Now, if the repressor is inactive, then the DNA bind domain shape is altered, meaning that RNA polymerase can start transcription. So it's going to, if the DNA binding domain shape is altered, it's not going to be able to fit in to this operator. It's not going to match up with this operator's shape, if that makes sense. So just know that repressors dictate whether RNA polymerase can transcribe genes. Now, the two operons that I'm going to be talking about are inducible operons and repressible operons. Inducible operons are always off because there is a repressor attached to it. The only way that RNA polymerase can start transcription on an inducible operon is if there is an inducer present that is going to change the binding domain's shape. So the lac operon is probably the most classic example of an inducible operon. The lac operon is responsible for breaking down lactose. The lac operon has its regulatory gene, and it also has its, I believe it's LAC-Z, LAC-Y, and LAC-A. Those genes are beta-galactosidase, permease, and transacetylase. And so essentially when lactose comes into play, so this would be lactose, right? This would be lactose. Lactose is going to bind to the repressor. It's going to change the DNA binding shape of the repressor, and it's going to RNA polymerase is going to be able to bind to the promoter, and it's going to transcribe these genes that are eventually going to break down lactose. So if lactose is the inducer and there is a repressor attached to the operator, the inducer is going to switch off this repressor, and it's going to allow RNA polymerase to transcribe these genes. That is called an inducible operon. So an inducible operon, like I said, is any operon that is always off, and it needs an inducer for it to allow for transcription. The other operon that I will be talking about is the repressible operon, meaning that they are always transcribing these genes, but they need a repressor to stop it. The classic example of a repressible operon is the trip operon, so tryptophan. So it just contains genes that code for enzymes that are needed to make tryptophan. So like when tryptophan levels get too high, tryptophan is going to act as a co-repressor, and it's going to bind to the repressor protein here, and it's going to change its shape so it can attach to the operator. Therefore, RNA polymerase is going to stop expressing these genes. So once again, quick overview, inducible operons are always off because they have a repressor attached to it, and they need an inducer, to switch off the repressor. Therefore, RNA polymerase would be able to transcribe these genes. A repressible operon is always on, and they need a co-repressor to change the shape of this repressor here so that it can actually bind to the operator and stop the transcription of these genes. That is just a quick overview of operons, and I hope you guys enjoyed.